What is a chromosome? In the nucleus of each cell, the DNA molecule is packaged into thread-like structures called chromosomes. Each chromosome is made up of DNA tightly coiled many times around proteins called histones that support its structure. What is a chromosome?
Chromosomes are not visible in the cell nucleus, not even under a microscope, when the cell is not dividing. However, the DNA that makes up chromosomes becomes more tightly packed during cell division and is then visible under a microscope. Most of what researchers know about chromosomes has learned by observing chromosomes during cell division.
Each chromosome has a concentration point called a centromer, which divides the chromosome into two sections, or arms. The short arm of the chromosome is labeled the P arm. The long arm of the chromosome is labeled the Q arm. The location of the centromer on each chromosome gives the chromosome its characteristic shape and can be used to help describe the location of specific genes.
The definition. A hybrid cross is breeding experiment between degeneration, that is, parental generation organisms that differ in two traits. Some examples are in this hybrid cross, a plant with the dominant traits of green pod color and yellow seed color is cross-pollinated with a plant with the recessive traits of yellow pod color and green seed color.
To understand the hybrid cross, it is necessary to understand the process of meiosis. You must have a clear understanding of the stages of meiosis. In the first meiotic division, the homologous pairs of chromosomes have aligned around them before being separated from each other. The separation of the chromosomes gives rise to the independent assortment of the chromosomes. In the second meiotic division, the four cells that are formed into haploid gametes.
First stage of parental cross. This shows a cell with two homologous pairs of chromosomes at the start of meiosis. The chromosomes have already replicated. The dominant allele for the gene for seed color is present on both members of one of the homologous pairs, and the dominant allele for the gene for seed shape on the other pair.
Second stage of parental cross. Pea plants grow from the seeds of the F1 generation are crossed. In the format of the cross, the phenotypes and genotypes of the parents must be shown together with the genotypes of gametes that can be formed as a result of meiosis.
Cross between members of F1 generations. Diagram 1. The homologous pairs have aligned in such a way that the chromosome with the dominant alleles will be separated together and the next stage in meiosis, and those with the recessive alleles will also be separated together.
Cross between the members of the F1 generation. Diagram 2. The homologous pairs have aligned in such a way that a chromosome with the dominant allele of one of the genes will be separated together with a chromosome with the recessive allele for the other characteristic at the next stage in meiosis. Each parent has produced the same four types of gametes. Fertilization is a random process with an element of chance. Therefore, any of the gametes from one parent can fuse with any one of the four types of gametes of the other parent. These possible combinations of gametes are best shown in Punnett squares. The possible gametes of each parent are set out along different axes, and the possible combinations of gametes shown within the squares.
Linked genes. Linked genes are found on the same chromosome and are transmitted together
unless crossing over has occurred during gamete formation, is a heterozygous parent. The parent characteristics will be present in the offsprings in approximately a 3.1 ratio of dominant to recessive. There will also be a small number of recombinants. This is important to increase variation within species.